think there's a lot of really exciting opportunities, and it was really on display actually amongst the 3 different talks. So our work is really kind of focused on it, using it as a tool for kind of neuropathology, a kind of more, we call it sort of genomic neuropathology, where you're really trying to go more in detail into the postmortem tissue than we've been able to before with traditional stains and molecular assays in situ. But you also have opportunities to really critically evaluate in vitro models. And our work informs that too. So because we have these definitions, these transcriptional definitions of dopamine neurons, we can now start to compare them to the dopamine neurons that are made in vitro as potential cell therapies for PD, but also as potential screening tools to identify drugs and genes that modify neuronal death in culture assays that people use in the lab. We can also use this same technology, as I'm saying, to basically tell us whether the models that we're making, the stem cell models we're making in the lab, are actually similar to the actual cells that are dying in the tissue. So I think there's a lot of really exciting opportunities there, and also as a readout for actual screens, because now you can ask, when you genetically modify the cells, you can ask again how similar they are, how different they are, whether they're turning on transcriptional pathways that are associated with neurodegeneration, or whether they seem more resistant to those kinds of perturbations. So I think it's a potentially just kind of a very ubiquitously used readout for many different kinds of assays in the lab, both within postmortem tissue, but also within animal and other laboratory models.